morning, everybody. So today we're going to finish the fuelization in response to food intake or feeding, right? And then we have a quiz. So I think we stop here, right? We finished the glucose metabolism after absorptive phase. That means 2 to 3 hours after meal when we eat, right? So what happened to the glucose in metabolism after we eat a meal, and then we started talking about the protein. So the liver, 60% of amino acids that actually initially from the diet are converted to carbohydrate and stored as the glycogen, right? So, and we also know that both carbohydrate and protein or amino acids are able to be converted to the carbohydrate, right? The glycogen is stored as a glycogen in the liver, and amino acids in the liver actually partially oxidize. The purpose of this to save the carbon skeleton, right? And prevent over production of ATP. And protein synthesis and urine synthesis and glycogen synthesis are all very active, right, in the liver during the fat conditioned absorptive phase, after we eat a meal. Because of a lot of protein our body needs to deal with, right? So the nitrogen metabolism will be also very active. So urine cycles we know is important way to get rid of ammonia, right, that initially from the nitrogen metabolism of amino acids. And again, I just want to mention that liver utilize mostly amino acids, right, as an energy fuel. Or preferred fuel for liver is, do you guys still remember, what is the preferred fuel for liver? For energy? Yeah, right, alpha Q acid, right, which initially from the metabolism of amino acids. So it's a little bit different from the other tissues, right? Most of the other tissues usually utilize glucose as a main fuel for energy. And this slide showing how amino acids or protein are metabolized after a meal, 2 to 3 hours after a meal, like in the absorptive phase. So, for example, we usually have get a 30 gram of amino acids from the diet. 10 gram of amino acids will go to the protein synthesis pathway, right? Will used for synthesis of protein and stored in the body during the absorptive phase. And 4 grams of amino acids, particularly like glutamine, glutamate, and aspartate, right? These are the most important amino acids that are utilized by intestine for energy, right? So about 4 grams of amino acids, the majority of amino acids goes to the intestine, glutamine, glutamate, aspartate. And 12 grams of amino acids from the diet will be metabolized in the liver. So for converted to glucose, right, through the gluconeogenic pathway, but stored in the form of glycogen, right? Does that make sense, right? In the liver, and some of the amino acids can be oxidized to produce ATP, right, through the alpha keto acid. And the urea synthesis is also very active, right? It's one of the way one of the pathway for protein metabolism, right? Or amino acid metabolism. And 4 grams of amino acid, particularly branch chain amino acids, will be metabolized in the muscle or maybe kidney, right, for ATP production. Here just sort of a highlight of the protein metabolism in the liver. The dietary protein, right? So 40 to 50% of dietary proteins are catabolized in the liver, and they basically converted to glucose and stored as a glycogen, right? So that is how dietary protein are metabolized in the liver during the absorptive phase. And how about the fat? We also get the fat, right, from the diet. So most of the fat in the meal is deposited in adipose tissue for later use, right, during the fasting. So the limited amount of fat goes to the liver and muscle tissue, right? So majority of the fat actually being delivered to the adipose tissue for storage. And the level of fatty acids are low in the absorptive state, right, 2 to 3 hours after meal, because so most of the fatty acids that we absorbed it from the diet actually goes to the adipose tissue and stored in the form of triglyceride, right, in the adipose tissue during fat state or absorptive phase. So that will remain the fatty acid level in the circulation low, right? So keep fatty acid level low in the circulation under the fat or absorptive phase. And here's the question: which lipoprotein transports dietary triglyceride from intestine to the adipose tissue? Do you still remember? Which? Chylomicron. Yes, chylomicron, right, is a main lipoprotein that is synthesized by intestine and transport the triglyceride from the diet, right, to the adipose tissue. And how are triglycerides taken up by the adipose tissue? How adipose tissue takes up triglyceride in the circulation, basically in the chylomicron and VLDL. Still remember the important enzyme that made by adipose tissue and secreted out of cells? Lipoprotein lipase, yes, lipoprotein lipase, right? It's very important. Try to remember this. Enzymes and lipoproteins, right? Okay, so and here show you the changes in plasma levels of glucose, insulin, glucagon in response to a meal, absorptive phase, right? 2 to 3 hours after meal. So you see that the glucose levels increase, right, after meal and reach peak level about 1 hour after meal, right? And in parallel, the insulin level is also increased, which makes sense, right? We know that glucose stimulates or triggers insulin secretion, right? So when we have a high glucose level that absorbed from the diet also increase the insulin level. So insulin and glucose level increase in parallel. But the glucagon, which is counter-regulatory hormones, the levels actually decrease, dropped, right? When we have a high insulin, high glucose, and glucagon levels is decreased. So try to remember the pattern of the glucose, insulin, and glucagon levels during the absorptive phase, right? Which hormones increase and which hormones decrease. And this slide tells us all the metabolic fuels levels, right? The levels of a metabolic fuels during the absorptive phase. So again, we expect the glucose level will be increased right after meal. So again, reach the peak level 1 hour after meal, and then the dropped, right, returned to the levels that before we are eating, right? And so amino acid levels, a slight increase after meal, but basically pretty constant, right? And triglyceride levels also increase a little bit, and lactate level actually increase a lot, right? After meal, the pattern kind of matches the glucose peak, glucose curve, right? So which also makes sense because glycolytic or glycolysis activities are very high or increase after meal, right? We want to utilize a lot of glucose, so a lot of tissue doing the glycolysis, right? Doing the glycolysis, so to release the lactate, particularly the red blood cells, right? Utilize a lot of glucose and release lactate, and also muscle tissues utilize glucose, and liver, right? But the fatty acid levels remain. Fatty acid, ketone body, the glycerol levels remain kind of low, right, under the fat condition or absorptive phase, which also makes sense, right? Under when we just eat and we have enough energy from the diet, we don't need to make ketone bodies, and we also don't need to release fatty acids for energy, right? So again, under absorptive phase, ketone bodies and fatty acids are low, right? The levels are low. Okay, now let's talk about the post-absorptive phase or fasting condition. So some kind of information that you need to know. First, the duration. So how we define the post-absorptive phase. So basically, it's the duration from 3 to 4 hours after meal to days of starvation, depending on how you're going, when you're going to the next meal or something like this, right? So the post-absorption phase, again, from 3 to 4 hours after meal to days of starvation. And what happens to the fuel utilization during this phase? There is a switch in fuel utilization, right? So fuel utilization switch happened during the post-absorptive phase. Two kind of switch. First is a switch of energy sources, utilizing exogenous source or dietary source to the endogenous source, right? The synthesized or stored energy. And the second type of switch is to switch utilizing the carbohydrate to utilizing fat, right? Two different type of a switch during post-absorptive phase. And also you need to know about the explanation for the fuel utilization switch. It's a Randle cycle, called a Randle cycle. So Randle cycle is a theory to explain the fuel switch during starvation, right? And last thing that you need to know during the post-absorptive phase is how does the body accomplish the fuel switch, right? We need to switch fuel, right? For example, during the fasting, the exogenous or dietary sources of energy is no longer available anymore. So then we need to switch to utilizing stored energy. So how our body can do this, right? The important thing is hormones. So lots of hormones like, insulin, glucagon, and catecholamines are the three main hormones, right, that regulates fuel utilization and fuel switch, right? And also substrate, malonyl-CoA and acetyl-CoA, right, the two important substrate that also be able to regulate fuel utilization switch, right? Again, hormones, insulin, glucagon, and catecholamine, and substrates, for example, malonyl-CoA and acetyl-CoA also be able to regulate switch of fuel, right, for utilization during post-absorptive phase. And again, this slide shows us the patterns or of the glucose, insulin, and glucagon levels, right, in the circulation during post-absorptive phase. So what we expected is that glucose levels dropped, right? If we don't eat meal, so the glucose levels dropped, and that also causes insulin levels decrease, right? So again, the parallel, glucose and insulin levels decrease, but glucagon levels increase. So again, try to remember the patterns of the glucose, insulin, and glucagon levels in the circulation during post-absorptive phase, right? Lower glucose, lower insulin, and higher glucagon levels, right? Okay, so this is something that we wanted to learn is changes in glucose utilization during, we put like absorptive and post-absorptive phase together, right? So divided these two phases into more detailed, like 5 different stages, right? So, for example, first stage or phase is the absorptive phase, mean 2 to 3 hours after meal, right? And then the second, third, and fourth, and fifth phase are considered post-absorptive phase, right? This phase actually defined by the time after meal or starvation. So if you look at the first phase 1, basically is absorptive phase, right? So the sources of energy, but here we specifically talking about the glucose, right? So in the phase 1, the main sources of glucose that are used by our body, this is from a diet, right? So exogenous glucose is the main sources of energy for our body in the phase 1 or absorptive phase. And phase 2 started from 4 hours after meal to 6 hours of starvation, right? So the phase 2 or state 2, basically utilize glucose from glycogen, right? So when we don't eat, so 4 hours after meal, then our body starts to utilize glycogen first for the energy, right? So glycogen in our body is completely depleted 16 to 18 hours after meal, right? So we use all of our stored glycogen if we don't eat till 16 or 18 hours after meal, after the first meal, right? So I want you guys to remember some kind of important time point, right? So 18 hours. So 4 to 18 hours after meal, our body primarily utilize glycogen for energy, right? The glucose from glycogen for energy. And after 18 hours, we don't have a glycogen store anymore, right? We use all of the glycogen. So gluconeogenesis then become important sources of glucose, right? So after 24 hours, gluconeogenesis is the only sources of glucose for our body, right? Couple time point, right? 18 hours is time point for glycogen depletion, and 24 hours is a time point for the gluconeogenesis becomes only sources of glucose for our body, right? But because this diagram only focusing on the glucose, right? We know we also utilize alternative energy, right? Fatty acids, ketone bodies. So fatty acids, like after 24 hours, our bodies start to break down fatty acids. Fatty acids level will become increased and also serve as important alternative energy for body. And ketone bodies start to increase after 2 days, right? Starvation for 2 days. After 2 days of starvation, right? Ketone bodies start to increase. Any questions about the timing of the starvation and how our body utilizes the different sources of glucose, right? Particularly. Okay, and this table sort of summarized the similarly, to the last slide, but in a different way. See, we again divided. We have 5 different phases, right, of the feeding or fasting, and how. So focusing on 3 different questions and try to answer. Try not to look at your notes, right? Look at the screen slides and see if you are able to answer the question. So first, phase 1, which is absorptive phase, right? 0 to 3 hours after meal. During this phase, what is the sources of blood glucose or origin of blood glucose? Diet, yes. So dietary or carbohydrate glucose is the main sources of glucose during phase 1, right? And what about a tissue using glucose? What type of tissue are using glucose? This is all tissues, right? This is a fat condition, basically, right? So all tissues will utilize glucose from the diet, right, as energy. Okay, so what about the major fields of a brain during this phase? Of course, glucose, right? Okay, and phase 2, which is earliest stage of starvation, right? So 4 to 18 hours after meal. So what is origin of the blood glucose? We just talked about in last slide. From which, of course, is not from diet, right? Starvation. So from the stored energy, yeah, glycogen, yes. Okay, glycogen. So hepatic glycogen, also hepatic gluconeogenesis. During this stage, the glycogen is a major source of glucose, but our body also started to making glucose through gluconeogenesis, although contribute less, right? So both glycogen and gluconeogenesis, but glycogen is more important than gluconeogenesis during phase 2, right? And which tissues utilize glucose? All tissues or limited some tissue? All tissues except liver, muscle, and adipose tissue at reduced rate, right? So because these three tissues, liver, muscle, and adipose tissues, take up a lot of glucose, right? So during the early stage of starvation, these three tissues, the uptake of glucose rate is reduced, right? The purpose of, to save the glucose for other tissue that for the brain, right, or red blood cell, because those tissues are very heavily reliant on glucose. And what about brain? Still utilize glucose, right, during the early stage of starvation as energy. Okay, now what about phase 3? 18 to 48 hours after a meal, right? So starvation for 18 or 48 hours. What is the sources of blood glucose? What do you expect during this phase? See the 18 hours time point, important time point, right? So past 18 hours, which pathway is very, carbohydrate metabolism pathway is very important to contribute glucose. Yeah, gluconeogenesis. We talk about after 18 hours, right? Glycogen is depleted, so we don't have a glycogen anymore. So we have to make glucose. So gluconeogenesis and then become more important, right? And what about tissues? Utilization of glucose, all tissues or limited tissues? Right, similar, right? Liver, muscle, adipose tissue continues to reduce the utilization of glucose, right? And brain utilization, the energy field for brain, glucose always, right? But what about the other alternative fields? Brain start to utilize during this phase? Well, still, right? So ketone bodies, we know, our body start to make ketone bodies when? How many days or how many hours? 2 days, right? So within the 48 hours starvation, brain will still utilize mainly glucose for energy, right? So now phase 4, starvation for longer than 2 days, 48 hours. So what are the main sources of glucose, blood glucose? What else, what are the other pathways that our body can make glucose? None, right? Breaking glycogen and making glucose through gluconeogenesis. So continued gluconeogenesis is the main sources of glucose, but the tissue that makes glucose is switched a little bit, right? So in the earliest stage, the liver is the main tissue that making glucose, and after 48 hours, that kidney become the more important tissue for gluconeogenesis by making glucose, right? And tissue, how about the tissues using glucose? Which tissues you expect to utilize glucose? Now, brain, red blood cells, right? Renal medulla, and small amount by muscle. So starvation for longer than 2 days, and lots of other tissues just shut down. The glucose utilization or slow down significantly, right? Glucose utilization, but brain, and red blood cells that are priorities, right, for glucose utilization. And what about the major fuels of the brain after 2 days starvation? Glucose, right? What about alternative fuels? Ketone body. So glucose still is an important fuel for brain, but brain also start utilizing ketone bodies for energy after 2 days. Okay. Okay, so last phase, the phase 5, and starvation for longer than 24 hours. Wow, it's a very long time starvation. So, and still gluconeogenesis, right, is the only sources of glucose in circulation. And brain, of course, is a priority tissue to utilize glucose. And ketone bodies now become more important energy fuel than the glucose, right? If at that long stage of starvation. Okay. Any questions? All right, now let's talk about the fuel utilization switch and regulation of switch. So changes in fuel utilization during post-absorptive starvation stage, we already talked about it. The 4 to 18 hours after meal, right? This duration is the phase 2, right? Phase 2. So blood glucose level is maintained mainly by breakdown of liver glycogen, right? And the brain utilize 50% of glucose, right? And utilization glucose by other tissues and is slowed down. I will try to save the glucose for brain and red blood cell and other important tissue for use. And muscle glycogen, I want to remind you again, muscle glycogen is not going to contribute to circulating glucose, right? Muscle breakdown glycogen just used for its own tissue will not release glucose into circulation. And the reason, if you still remember, which enzyme is missing in the muscle? Glucose 6-phosphatase, right? Okay. And after 18 hours starvation, the liver glycogen is depleted, right? We use all of the glycogen in the liver. So 18 hours is the time point, right, for glycogen depletion. So now what happens after 24 hours? Basically, we mentioned earlier, we just want to go through it again, right? The gluconeogenesis now become more important sources of circulating glucose, right, compared to glycogen, because glycogen is already gone, right, after 24 hours. And the only substrate for gluconeogenesis during this time period are amino acids, lactate, pyruvate, and glycerol. I guess everybody knows, right? The acetyl-CoA is not able to make a glucose, right? It's not the precursor of a gluconeogenesis. So the other, a lot of glycolytic tissues, the tissue that utilize glucose, will not completely oxidize glucose, but partial oxidize glucose. The purpose of this is to save carbon skeleton, right, for the resynthesis of glucose or gluconeogenesis. So if you still remember, we have two important gluconeogenic cycles, Corian cycles and alanine cycles, right? Both cycles, they use different precursors. And during this stage, the brain is the only tissue that still completely oxidize glucose, right? Okay. So again, in addition to glucose, we know our body is able to utilize alternative energy fuels, right, including fatty acids, ketone bodies. So after 24 hours starvation, the fatty acids now become important or energy fuels for body, and 48 hours or 2 days later of starvation, ketone bodies are made, right, by the liver and provide, again, important alternative fuels for brain and adaptation muscle, kidneys. Okay, so here is information about the plasma concentration of energy fuels during prolonged starvation. So if you can see that the ketone bodies, right, raise significantly, you see the ketone bodies start to increase 2 days after starvation, right? And increase really a lot and can maintain the very high levels during, this days of starvation. I'm not sure that we are still able to survive for that long period of starvation. But anyways, that, keep a very high level for a very long time, right? And glucose dropped, right, significantly, and also, let's see what else we decreased. The increase, the fatty acids and triglyceride and amino acids will all increase. But during this very long starvation, the lactate and glycerol levels maintained very low, right? So I listed here during this very long starvation period, the glucose, amino acids, and lactate were significantly lower, right? But ketone bodies, fatty acid, triglyceride level were significant increase, high. So the question here, why lactate and amino acids are decreased in the circulation during the period of starvation? Why? Because we know lots of tissues, right, try to spare glucose and only do the partial oxidation of glucose, right? Well, we expect the lactate should produce a lot, right? But why lactate level or glycerol level are low? Glycerol we also expect to very high, right? Because we break down triglyceride, release fatty acids and glycerol. Why these two compounds maintain a very low level during the starvation? Any idea? Where do these compounds go? Yeah. Yes, excellent. Because gluconeogenesis are very, very needed, right? Urgently needed during this long period of starvation. So lots of lactate and glycerol are used for gluconeogenesis to make glucose. So that's the reason why we have a very low levels of lactate and glycerol during the starvation compared to the fat condition, right? Does that make sense? Okay. All right. Now, let's talk about a switch. The people use so-called random cycle to explain a fuelization switch. So, so basically, this series is the Randle cycle is a metabolic process involving the competition of glucose and fatty acids for substrate utilization in the starvation. And this theory can also be applied to the type 2 diabetes insulin resistance, right? So in other words, that in general, our body utilizes glucose and fatty acids. They are kind of compete. So, for example, if we utilize more glucose, the fat utilization will be inhibited, or vice versa. If we utilize more fatty acids, for example, during the fasting condition, then glucose utilization will be reduced. So the kind of competition exists between glucose and fatty acid utilization, right? So the Randle cycle is also called a glucose fatty acid cycles, right? And the purpose of these cycles basically is the effect of a glucose sparing, right? So fatty acids has a glucose sparing effect. That means when we utilize more fatty acids, we can save glucose, right? Inhibit glucose utilization to save glucose. So if you look at under the fat condition, when we have lots of blood glucose, right, from the diet, we stimulate insulin release and inhibit glucagon